a broad sense, my lab is interested in two intersecting questions. The first is understanding the neural circuitry that supports our ability to make decisions about when and how to invest effort in things that we want. And we tend to focus, just by contrast to sort of what we were saying in the prior talk, we tend to focus on very temporally local decisions. Like, what am I going to do with my effort right now? A good example is a gathering like this. All of you at some point this morning were presumably comfy in bed, and you're thinking, is it worth the effort to go to this meeting, to hear this Treadway guy? Like, we'll find out. But you made that decision. And how the brain does that, what's the neural circuitry involved, that's what we're interested in understanding. And one of the reasons we're interested in this, apart from just being an interesting question, is those types of sort of simple everyday decisions that many of us often take for granted are in fact heavily impacted in a variety of forms of mental illness. Most prominently, major depressive disorder, and I'll talk a little bit about our work in depression, but it's also a common feature in a wide range of other conditions, including anxiety disorders, addiction, and even schizophrenia. In the context of depression, what I'm describing is the difficulty making decisions or finding the motivation to do everyday tasks, getting out of bed, taking a shower, going to work. It's referred to as anhedonia, which is defined in the DSM-5. The DSM is sort of the Bible of psychiatric disorders. It's defined in the DSM as a lack of interest or pleasure. So people can kind of be diagnosed with anhedonia because they say, well, I just don't feel motivated to do things, or I do feel motivated, but when I do, I don't enjoy it. And that's sort of all treated as equivalent towards the DSM. And this, I have argued, and others have joined in, this is actually a very problematic definition if you care about understanding the underlying neural circuitry. The reason for that is there are distinct neural circuits, which I'll be presenting on today, that support wanting something or feeling the motivation or the willingness to work for something, as opposed to actually enjoying it. Mm-hmm. These circuits talk to each other. It wouldn't make sense if they didn't, but they are dissociable, and that means in the context of illness, they can separably break down. And people may actually have different problems that you need to tackle. This is a complicated figure. The takeaway here is it's complicated. We'll try and break this down a little bit. So how did we begin as a field to figure this out, that these were separable? This work kind of began in the mid-1990s, where there's a lot of focus on that time in studying dopamine, and the guiding idea was that dopamine was about pleasure. Dopamine made you feel enjoyment. And early cracks in that theory started to emerge from studies using a paradigm like this one. This is a Skinner box for rats, and in the box, the rat is trained up to know that it can press a lever 5 times and get a sweet pellet, or it can ignore the lever entirely and just freely access this sort of standard lab chow. So preferred food reward, but you got to work for it, or you can just eat what's available. And healthy rats you put them in here, they for the most part ignore the chow, and in a 30-minute testing session, they'll make about 1,500, 2,000 lever presses. The lever is about half the size of the rat, so imagine sort of like a whole body exercise, right? So that's a fair amount of work. They're working hard. And what John Salamone at University of Connecticut and others have shown is that if you block dopamine with various drugs, this one's using a VAT blocker called tetrabenazine, you get this dose-dependent decrease in lever pressing, so you work less and less, and importantly, you get a compensatory increase of the free food intake. And this is critical because what it's telling you is blocking dopamine doesn't get rid of your appetites, right? It doesn't just, you don't want to eat anymore. What it tells you is it's selectively ablating your willingness to work for a preferred reward, right? And other control conditions have been done. I don't have slides for them, but if you take away the effort requirement, even rats on high doses, they'll eat sweet pellets if they don't have to work for them. If they're freely available, it's not like they got confused that which do I prefer. That's one thing. Around the same time in the 1990s, Ken Berridge at the University of Michigan was going after this liking question. And this is very difficult in animals because you really can't ask them like, hey, how much do you like that? But he did something very clever, which is to recognize that across mammalian species, there are conserved facial reactions to pleasurable stimuli, like sweet tastes. And you're seeing, it's a little hard to see here, but a rat sort of licking its lips. Monkeys do the same thing, and newborn babies will do the same thing. And I don't have it, but when my infant daughter was born a few years ago, I tested this out with some breast milk, and it works, you know. So these are reliable indicators of sort of, I like something. And what Ken found is if you manipulate dopamine, you get no change in this. Dopamine, you block dopamine, doesn't go away. You increase dopamine, doesn't get more. What drives this is opioids. All right, and he's done a lot of studies in this. This is data from one study where what you're seeing is he's doing these microdoses, micropipettes of mu opioids in different brain areas, and you're seeing the increase in these liking reactions. And mostly mu opioids give you a big increase. It is region specific, so there are some spots in the brain, these cool spots, where you get a reduction. And you get no change in willingness to work for rewards when you give opioid drugs. So you get this real nice dissociation. As an aside, I don't have a figure for it, but more recently, Kent has mapped stimulation for either increasing wanting or increasing liking throughout the brain. And the bad news, if you've ever tried to successfully be on a diet or an exercise program, is the brain has many, many, many more neurons that will increase wanting when stimulated than increase liking. We are wired up to want more than to enjoy. Okay? So, but this is in rodents, and I wanted to see, my sort of career began trying to see, could we show some of these phenomena in humans as a way of understanding human circuitry. So, for example, if loss of dopamine in the rodent striatum leads to this reduction in lever pressing, maybe if we increase dopamine, we can increase willingness to work for rewards in humans. So about 15 years ago, I developed a task that was sort of meant to model the animal task that I showed you, the lever pressing that showed sensitivity to dopamine. It's a very simple paradigm. We call it the effort expenditure for rewards task, or just the effort for short. And in this task, people are given a series of choices where on each trial, they're told you can either do a low effort task for a chance to earn $1, or you can do a high effort task for a chance to earn somewhat more than $1, between $1.25 and around $4. That varies on every trial. And you're also told that you have a certain probability of winning, so it's not a guarantee. And we did that because, in life, putting in your effort doesn't usually guarantee you reward. It's probabilistic. So you make your choice. Then the effort in our hands is rapid button pressing. The easy task, or the low effort task, it is 30 button presses in 7 seconds with your dominant hand index finger. And the hard task is 100 button presses in 21 seconds with your non-dominant pinky finger. I have done this task myself more than I care to admit, and I can tell you it is effortful. Especially, and you're told also, importantly, you have 20 minutes to get to choose whatever you want. So you don't get through the task quicker if you make different types of choices. You're there for 20 minutes. And our primary dependent variables of this task is just A, the proportion of hard task options that were selected. And then we can also do some computational modeling. It's not a big part of this talk, where we can estimate your subjective value point for how you value the effort, and we can look at sort of your effort discounting parameters. Okay, so the first thing I wanted to do to kind of translate what had been seen in rodents is we did a double-blind, multiple-dose amphetamine challenge study on this task. People came in for 3 visits, placebo, 10 milligrams, 20 milligrams of D-amphetamine. And we found, this was about 15 years ago as well, in a small sample, 17, but all within subject, crossover, double-blind, that you did get a dose-dependent effect where giving amphetamine increased willingness to work for rewards. About 5 years ago, we set out just to replicate this. Everyone likes replication. We did a larger sample, and we basically got the exact same effect. This was done in collaboration with Megan Wardle at University of Illinois, Chicago. We would look at in these studies, like, does the change that amphetamine induces in terms of your effort, does that at all correlate with how much you are reporting you're liking the drug experience? And we don't see any relationship, but these are pretty small samples for that type of analysis. More recently, we replicated the study in a sample of almost 100 participants. This is now a single dose methamphetamine as opposed to D-amphetamine, which is a little more potent. We got the same effect of effort, and I don't have a figure for it, but the summary in the abstract, the behavioral effects were not related to drug liking or drug-induced euphoria. So we got, people have some euphoric response to drugs like amphetamine, but that was not at all sort of correlated with how much it changed their motivation. So again, suggesting potentially separable systems. So what about liking? A few years ago, a collaborator of mine at Emory, Phil Kragel, and I set out to see if we couldn't map liking and see if the circuitry of liking was distinct in humans as compared to the circuitry of wanting. And here we took kind of a different approach than the behavioral pharmacology approach. We looked at a large sample of available neuroimaging data sets. So we have a total of almost 500 participants across a variety of studies. And we took studies that looked at 5 different domains of pleasurable experience in the scanner. So playing pleasant music, showing pictures of appetizing food, showing short videos of positive interactions like babies playing with puppies that people tend to like, showing some erotica images that tend to engage pleasure systems. And then we contrasted them with studies of cognitive control in red, pain in green, and negative affect in blue. And here we use a multivariate imaging approach where we're basically searching this corticostriatal network, so only the areas in color, those were areas we looked at. And we use a multivariate machine learning approach to say, what are the weights on all these voxels that can classify the pleasure studies as different from all these other types of studies? And this is the sort of network that you get. Now, with this type of machine learning approach, it's prone to the real risk of overfitting, like just something specific to your data set. So then once we had this, what's often called a neural signature, we then applied it to several studies that were not part of the training data set. to see if we could classify. And we were particularly interested in, would this be selective to sort of pleasure versus just reward, right? Money is an abstract reward. And so we looked at humorous cartoons, pleasant versus neutral tastes, and then 2 monetary reward tasks. And what you can see is we get much better classification for the 2 sort of more sensory pleasure, even humor and pleasant versus neutral taste as compared to these 2 monetary reward tasks. Now, is this related to opioid function? We don't have quite direct evidence, but we do have some that I think is worth noting. So within the last 10 years, the Allen Brain Atlas has published whole brain gene expression maps that show you the localization of particular receptor subtypes. So here we have both the maps of distribution, spatial distribution of expression for the dopamine, 5 dopamine receptors, and then the different, the 3 primary opioid receptors. I should note, when people talk about opioids being addictive or opioids drive pleasure, they're talking about mu opioid agonists. Kappa and delta are actually, can be mildly aversive, and they have nowhere near the same effect. So what we wanted to see is, were the sort of weights in our signature, did they show some similarity to the distribution of mu opioid as compared to some of these other receptor distributions? And that is indeed what we saw, strongest association for mapping onto mu opioids. This is this regression coefficient uses spatial regression to see basically the similarity between our signature maps and the spatial distribution of these receptors. Interestingly, we also saw something for the dopamine DRD2. We don't know exactly what that means. It may be that there's just a lot of co-localization. But the biggest effect was for the mu opioids, which was also significantly stronger than the other opioid receptors. Finally, we took the signature into a study that had administered naloxone, which is an opioid antagonist. And we see a significant, a larger impact in our signature's similarity to the results of that study on the effects of naloxone for erotic images, but not for monetary reward. So again, we're sort of getting this separation of real sensory pleasure from sort of more abstract monetary reward. And then finally, a separate group led by Tor Wager did use a very similar approach to classify monetary reward. And so we looked to see, like, well, would these behave differently? They should, and indeed they do. So here in the pleasure signature, you've seen this before, we do pretty well with the blue humor and pleasant tastes, but not so good with large rewards. Here the reward signature does very well with large rewards and not so well with the others. I will say, you may be wondering, well, why does it do bad with large versus small reward? This study comes from a study of effort-based decision-making. So actually they were looking at rewards, but knowing they would have to work for them. I'll come back to that, why that looks the way it does, a little later on. So just kind of summarize part 1 here. Effort-based decision-making is sensitive to dopamine manipulations, and we are able to sort of pull out distinctions in liking versus wanting circuitry that seems to, at least in part, reflect a translation of what's been shown more experimentally in animal models. Okay. So I'm going to shift gears a little bit. I do mostly laboratory work, as you've seen, that is very controlled and not very not necessarily very ecologically valid, right? That's really a limitation, right? No one really experiences real hardship in life because they're not willing to press a lot of buttons with their pinky fingers, right? That's not a problem. So more recently, we've been trying to see how this sort of separation of deficits in motivation and pleasure may play out in daily life. And we use ecological momentary assessment, basically smartphone surveys that are sent out to people and they complete multiple times throughout the day. So this is recent data. This hasn't been published yet. This is sort of my first time presenting it. We've aggregated across 3 different studies 155 participants, about half of whom are they go through a clinical interview and they're confirmed to be healthy controls, no psychiatric history, and 50 with depression and anhedonia, and then a smaller group with anhedonia but don't have full-blown depression. And they did EMA surveys between 4 and 8 weeks, 6 times a day, every other day. Okay? The surveys are brief. They only last about 3 to 4 minutes, and we have had fairly good compliance with our protocol, usually above 80% compliance for both depressed patients and healthy controls. I'm happy to talk about how we do that. And what we ask people to do, very simply, is after, or beginning with the first one of the day, we ask, here's a list of activities. What are you going to do in the next 2 hours? You're going to go to work, you're going to study, you're going to exercise. You can pick up to 4 things you might do. And this isn't an exhaustive list. There's more you can scroll down. At the end, you can select other if it's not on the list. But most things are on the list. Only about 10% of survey responses have other. Okay? And then once you've selected your activities, we just ask you with these little sliders to rate how rewarding do you think it's going to be? How effortful do you think it's going to be? Okay? And then we ask those kinds of questions. After the first survey of the day, so we get to so after survey 1, there's 5 more. At those surveys, we also ask you to look backwards. You said you were going to do this activity. How effortful was it? How rewarding was it?
Did you ask, did you do it?
Yes, yes, absolutely. Absolutely. We did ask, we first asked, did you do it? Yes, yeah. Thank you. And then we also ask you to sort of rate your momentary affect using items from the positive negative affect scale. Okay? Now we're interested, there's been a lot of attempts with self-report measures to kind of pull out motivational versus consumatory deficits, which I'll refer to as motivational anhedonia versus consumatory anhedonia. One that I have felt does a particularly good job is the Mood and Pleasure Questionnaire developed by Anne Kring and colleagues. So we use this as our sort of proxy, or our way of identifying people who might have more motivational deficits and more consumatory deficits. And our question is like, are these really separate things in daily life, right? One other thing that we did is for our activities that people were selecting from, we recruited a separate sample of 100 individuals to provide their ratings on on average, how rewarding and effortful is this activity, just to have sort of an objective basis as a comparator. Okay. So like I said, this is my first time presenting it. The presentation is a little less polished than usual, but first looking at how people rated on average their effort as a function of having motivational anhedonia with consumatory anhedonia also in the model, along with sex, age, and the study sample that a participant was in. And what you see is that the higher your motivational anhedonia, the fewer number of objectively effortful activities you plan for. So kind of makes sense. Interestingly, if you're higher in consumatory anhedonia, those individuals are actually on average doing more effortful activities, right? So lack of effort is not their problem. If anything, they're working a little harder, right? And I don't have the plots for it, but if I look at, instead of planned activities, completed activities, very similar results. Which in the data, generally, most people did what they planned most of the time. But we always check that. So motivational anhedonia is selectively associated with effort avoidance, while consumatory anhedonia is not. Now, what if we look at the reward of activities? Okay? Here, individuals with motivational anhedonia are actually pursuing more rewarding activities, right? And I think this tends to reflect a preference for low effort, high reward activities. Watching TV, social media. These tend to get rated as more rewarding, and this is what they're spending more of their time doing. And consumatory anhedonia, again, just the opposite. So, kind of, construct validity here. Consumatory anhedonia is selectively associated with a failure to engage in objectively rewarding activities, while motivational anhedonia is not. There's a lot more we've done with this data. I'm just going to show one other set of analyses for today. So when we ask people to rate their current affect after that first survey, there's a number of potentially influencing factors that we've collected. The level of effort and reward of what they just did, right? I just took this exam, it's over, blah, blah, blah. It was way harder or way easier than I thought. There's potential prediction error, right? So I thought it was going to be really easy and it was way harder. That's a prediction error. And then there's looking ahead, what am I doing next, right? So my current mood could be more influenced by what just happened or where I'm going. It's going to be a mix of both. So we've been testing out wanting to see how those dynamics are moderated by levels of motivational and consumatory anhedonia. So these are linear mixed effects models with random intercepts. And I'm just going to highlight two results that I thought were particularly interesting. So first of all, looking at negative affect, moderated by motivational anhedonia. The solid lines are planned activities. The dotted lines are the prior activity. Okay? So for whether you're high in blue or low in red, your prior activity doesn't drive your negative affect that much. Your planned activity drives a lot more negative affect for high motivational anhedonia. They anticipate effort with a lot of negativity. Okay? And I should stress, unlike the prior results, we're now looking at their subjective ratings of the effort of the next activity. Okay? Now, I'm also a clinical psychologist trained in sort of cognitive behavioral therapy, and one of the things you do with people with depression is you try and get them to do stuff. And it's because of this, because they're anticipating effortful things. I don't like it, I want to avoid it. And the idea is, if they just do stuff, they'll find it's better than they expected. And so we were interested in that phenomenon, and indeed, looking at positive affect, again, solid is planned, dotted is prior. Sorry, the colors are now different. So red is now high. Just ignore planned for a second. Prior activities, if I just did something effortful, it actually boosts my positive affect if I'm high anhedonic, right? So the thing that when I look forward to it, drives my negative affect is actually the thing that really helps me when I do if I actually get myself to do it, okay? So we're excited about this as a nice demonstration, I think, that really hasn't happened in the literature of the principle of therapies like behavioral activation therapy. And what's interesting is you don't see this result for high consumatory anhedonia. So it again suggests, you may have differential treatments that are needed. Okay. So now I'm going to take a bit of a turn because I did want to talk a little bit about how we use this, how we think about using this kind of information to understand pathophysiology, like where does this circuitry go wrong and what are ways we can intervene. And so I'm going to give a little background here, just bear with me. Inflammation and depression has a very long history, going back to the 1980s when people started observing that animal behaviors, when they're highly inflamed, they look very similar to the animal models of depression. The animals sort of huddle up, they don't want to work for anything, and when we get sick, we often do kind of similar behaviors. We stay in bed, we're kind of irritable, we don't really want to interact with people, we're not enjoying things. So people started to wonder, gee, could immune engagement be playing a role in depression? And beginning in the early 90s and consistently in the literature across multiple meta-analyses, you see that yes, patients meeting criteria for major depression show elevations in inflammatory markers in blood samples. And here's just one recent meta-analysis, but there have been many. The green arrows indicate the most commonly found inflammatory markers, which include TNF and C-reactive protein. I apologize for sort of the alphabet soup you get. And what this study also did, which was a really nice addition, rather than just showing, yeah, on the mean you find these elevations, they looked to see, okay, in each of these studies, what proportion of patients are showing elevations? Like, is it everyone with depression has increased inflammation, or is it just a subset in each study? And so now this is the proportion of patients who showed an elevation of a C-reactive protein greater than 3, which is CDC guidelines for defining high inflammation. That's where that cutoff comes from. And what they show is it's around 26%. So it's a subgroup. The vast majority of people meeting criteria for depression have no problem with inflammation whatsoever. So if you go around just prescribing, aspirin or Advil to people for depression, it's probably not going to do very much. For my money, what kind of really convinced me to get excited about this were a set of studies serendipitously that kind of went the other way. These are studies that examine the effects of a drug called interferon alpha, which causes a massive immune inflammatory response. They used to be used to treat certain types of cancers and hepatitis. And what researchers found repeatedly is that a subset of people receiving this drug would develop a serious depressive episode. And here's one analysis from a New England Journal paper. So at baseline, 100% of these patients had no psychiatric history. They were pre-screened, and they were all being treated for hepatitis C. After about 12 weeks on interferon alpha, about half of them were significantly depressed. What's interesting about this, it's really interesting that giving a pro-inflammatory drug is sufficient to induce depression. It is also interesting that about half these people don't have that response, right? It's not that inflammation causes depression. It's some people appear to have a biological vulnerability to the effects of inflammation. And what that vulnerability is, that is the million-dollar question in this field. And this is just showing that this was a large sample study from the UK Biobank showing that the effects of inflammation on depressive symptoms, it's not all depressive symptoms, the ones that come up most strongly are felt everything was an effort, and could not get going, loss of energy. Okay? So what we've been wondering in the last 10 years is inflammation suppressing dopamine? And increasingly there's evidence that it is. So this, in that same paradigm I started with, you saw dopamine blocking drug reduce willingness to press lever for reward. Now you're looking at a dose of a inflammatory cytokine and you get the exact same effect. If you measure dopamine when you give an inflammatory cytokine in white here, you get a clear reduction. My collaborator at Emory did this same type of study in humans. So this is F-DOPA imaging, a PET measure of dopamine synthesis capacity, and you get substantial reductions after 4 weeks on interferon alpha therapy. Okay? This is a table from a review that we did a few years ago, basically just showing from rats to humans. It's not a huge literature, but the results are very consistent. You give a pro-inflammatory drug, dopamine is reduced, and willingness to expend effort is reduced. And I'm a little short on time, so I'm just going to jump to this last study I want to present. This is a clinical trial we did. We wanted to test this idea of an inflammatory subtype by examining whether an anti-inflammatory drug, a powerful anti-inflammatory, infliximab, which is a TNF antagonist, whether this could reverse motivational deficits in patients who were both depressed and had evidence of high inflammation. Okay? And so this was our design. People came in, they went through extensive screening. They then did some neuroimaging and some effort tasks like the kind I presented earlier. Then they were randomized to infliximab or saline using double-blind protocol. And then after 2 weeks, we tested them again. The task we used it's a little different in that it's the same basic idea. You want to work more or less, but it has different levels of effort because we wanted to see sensitivity to that. And what I do think is important is they're doing this during an fMRI scan. They make their choices of what is the effort I'm committing to do in the scan, and then they actually do the effort outside the scan. And so think back to that, when I am thinking about effort I have to do, you get that strong negative affect. This is that sort of anticipatory effort. And at baseline, we saw in this sample that, if you had higher baseline anhedonia, you did make fewer effortful choices. Our pre-registered hypotheses were we thought the drug would suppress inflammatory markers. We had several self-report questionnaires that we thought would be sensitive to motivational anhedonia. We had this task behavior, and then we had some hypotheses about how brain activity would change. So to quickly go through these, yes, our drug worked. So the placebo, their CRP didn't change from baseline to day 14, and infliximab group it did. This is a plot of effect sizes for that group by time interaction. The cytokine that had the biggest effect was a TNF marker. That's the pathway the drug targets, so that makes sense. So we use this as our proxy of target engagement, because a single dose of a drug, any drug really, will have varying effects. It'll work more effectively for some people than others, right? So looking at our self-report measures, it was kind of a mixed result. We did get some strong effects for infliximab relative to on 2 of the measures, but we also got a reasonably strong placebo effect. Infusions usually get stronger placebo effects because it's sort of it's a bigger deal. And I think with only 2 weeks, we may not have had enough runway for the placebo effect to wear off. So even though the effects are nominally stronger in infliximab, we didn't get a significant group by time interaction. on this measures, on these sort of self-report measures. For our effort task, so this is the proportion of options they accepted at each level of effort. It's again rapid button pressing, but now a sort of percentage of your max rapid button pressing. See no differences between the two groups at baseline. At 2 weeks, however, we did get a group by time interaction and a group by time by effort level interaction, such that the drug selectively increased patients' willingness to work for reward at the two most effortful levels. This is just a subjective discounting curve, kind of another way of modeling the data that sort of shows pretty much the same thing. You get less discounting of reward by effort level in the Inflix group. So then we wanted to know, well, was this related to how effective the drug was? So we looked at whether this change tracked with suppression of TNFR2, and we found in both cases it did, or both of these metrics, I should say, and you got significant mediation between drug assignment and behavioral change as a function of how much change in TNFR2 there was. I'll just show one or two main brain results. So we were interested in fMRI signal in the striatum, which has sort of the richest amount of dopamine. Our hypothesis, again, is that inflammation is suppressing dopamine. If you block that inflammation, you should see a dopaminergic rebound. We didn't get a direct effect on amplitude of the fMRI signal, which is not a direct measure of dopamine, but we did see that the ventral striatum could, using a sort of multivariate classification, could classify who got placebo or drug. So change in this signal for the ventral striatum held information of who got what condition, what treatment, and that was not true as you moved into other parts of the striatum. Lastly, we used that monetary reward signature I showed you, and we wanted to see if this monetary reward signature sort of capturing, like, is the brain in more of a win-like state or a loss-like state. And so we wanted to see, would this be sensitive to changes of the drug? This is just showing in a validation sample of healthy controls, because we didn't know if it would work on our task. If you look at high subjective value trials, so where the sort of the effort is worth the reward, it looks about kind of like a 0 point on this signature, but when the reward is not worth the effort, it actually looks like a loss, okay? And this is just showing that not all signatures are sensitive. And so what we found is that changes, as your anhedonia improved, as it went down, you started to look more win-like on this signature, and as your willingness to work for rewards went up, you started to look more win-like on this signature. And there was no association with the pleasure signature. Again, sort of suggesting we're targeting separate pathways. So basically, we do think this provides some validation for an inflammatory subtype, and we think that in broad terms, there's growing good evidence for separate motivational and consummatory deficits that may respond to differential treatments. So with that, I'll thank the many people who've helped with this work, and thank you all for your attention.